Good afternoon to one and all. So, myself, Dr. Damodar Chari, Assistant Scientific Officer, Microbiology, NIPHM. So, today's session is production and use of biofertilizers in organic farming. So you know about different plant requiring nutrients. So essential plant nutrients are required almost 16 or 17. So among all macronutrients are there: nitrogen, phosphorus, potassium. So these nutrients are generally supplemented through chemical fertilizers, and biofertilizers also there for supplying these macronutrients. And plant growth promoting rhizobacterial inoculants also is there for supplementing these nutrients. And secondary nutrients like sulfur, calcium, magnesium, majorly these nutrients supplemented through chemical fertilizers. And micronutrients like boron, copper, iron, manganese, molybdenum, zinc, and selenium. These nutrients are chemical fertilizers are available, and seaweed extract also using for supplementing these nutrients. And plant growth promoting rhizobacteria also solubilizing different micronutrients. So these formulations also using for supplementing these nutrients. And other nutrients like carbon, hydrogen, oxygen, these nutrients are generally derived from the nature. So why fertilizers are required? Generally soils may be naturally low in nutrients, so deficient due to nutrient removal by different crops, particularly high yielding varieties are removing nutrients from the soil. So soil required some nutrient content. So this compensated by application of fertilizers. So generally fertilizers, broadly chemical fertilizers are there, biological fertilizers are there. So chemical fertilizers generally using in conventional farming, and biological fertilizers like biofertilizers, organic manures, like these biological fertilizers are using organic farming practices. So what are the advantages of chemical fertilizers? So generally chemical fertilizers are immediately soluble, and they are readily available to the plants after solubilization, and direct effect and fast effect is there with the chemical fertilizers. But these under subsidy farmers are getting these chemical fertilizers, and they are popular with the farmers. So more promotional activities are there and subsidies are there, so they are popular with the farmers. And this nutrient content also high in the chemical fertilizers, and small quantities are enough required for crop growth. So disadvantages are there, a lot of disadvantages are there using with the chemical fertilizers or agrochemicals. So generally negative effects are there, such as leaching losses are there, and pollution of waters. So accumulation of more nitrates and phosphates in the aquatic conditions, it will create eutrophication, more algal growth, and water pollution water pollution is there with this accumulation of nitrates and phosphates. And also important beneficial microorganisms in the soils and beneficial insects, and their number will be destructing and reducing. And crop application of more content of these wide amount uses of these chemical fertilizers, crop will be susceptible to the disease attack, and problematic soils are creating acidification, alkalization of the soil. And fertility loss, finally fertility loss is there, so that cause the irreparable damage to the soil, overall soil ecosystem. Sometimes oversupply of nitrogen leads to softening of plant tissue, resulting in increased susceptibility to the diseases and pests. And decomposition. Sometimes these fertilizers, their nutrients, the high application, they enhance the decomposition of the soil, and degradation of the soil structure may be happening. And nutrients are easily lost from the soils through fixation with other nutrients, and leaching and gas emission, and can lead to the reduced fertilizer use efficiency. So why we need revival of soil health by our good agricultural practices, particularly the application of certified organic inputs like biofertilizers, efficient biofertilizers, novel biofertilizer inoculants, organic manures, crop rotation, like this. With certified organic inputs, we will revive the soil health. Whereas chemical fertilizers, the soil health will be disturbing, rhizosphere health will be disturbing, long-lasting the effect will be there. So this with certified organic inputs, we will improve the organic carbon content, microbial number, beneficial microbial number and activities, and humus content. Soil health will be increasing, soil fertility will be increasing, crop productivity will be automatically increasing. So coming to biofertilizers, so biofertilizers generally the biofertilizers lab establishment under fertilizer control order guidelines. So this is the definition. So biofertilizers are the product. So they are solid form or liquid form, any other form. These formulations are containing effective microorganisms, efficient microorganisms of different agricultural beneficial microorganisms, their strains. So these strains are useful in terms of nitrogen fixation, or phosphates or phosphate solubilization, or other nutrient mobilization or mineralization activities, and also using as a composting inoculants also. So these organisms, while we are applying these formulations as a seed treatment or soil application or any other method of application, they are increasing the productivity of the soil or crop. So this is the biofertilizer definition. So why biofertilizers are essential? Because of to revive the soil health and a living soil concept which support to a soil sustainable production system. The soil environment ecosystem need to made congenial for living of the useful microbial population, responsible for continuous availability of nutrients from the natural resource. To increase the soil quality and soil health, the biofertilizers are essential. So continuous supplying of nutrients for sustainable agriculture system, the biofertilizers are promoted and required. So biofertilizers generally capacity to reduce the up to 50% chemical fertilizers and enhance the 20% yield. So these improve the soil fertility, improve the reclamation of soil, help in reclamation of soil, safe to environment and health, maintain natural resources. So indirect benefits to biofertilizers are helping in developing the agri-entrepreneurship, save in foreign exchange, food and nutritional security, check rural migration, conservation of natural resources. So next, different types of biofertilizers are available. These are nitrogen fixing biofertilizers, phosphate solubilizing microorganisms, phosphate mobilizing microorganisms, and zinc solubilizing bacteria, potassium releasing bacteria, sulfur oxidizing bacteria, and different PGPR bacteria biofertilizers and compost inoculants are there. So coming slides, I will explain about different types of biofertilizers and their significance. So nitrogen fixers. So you know nitrogen is an element. It is essential for different all forms of life, and it is vital element for plant growth and productivity. So you know about almost 78% of the atmosphere containing gaseous form of nitrogen, N2. So that is unavailable to the plant roots, absorbing unavailable form to the plant roots. So these unavailable form will be fixing by different soil microorganisms. That microorganisms are known as nitrogen fixers. So these nitrogen fixers are involving important process, that is biological nitrogen process, biological nitrogen fixation. So this biological nitrogen fixation, the important enzyme which is present in the bacteria, that is a nitrogenous enzyme, it is involving. So this, after nitrogen fixation, the gaseous form of nitrogen is converting into ammonical form. So that is the main important reaction in the soil. So this biological nitrogen fixation. So the nitrogen cycle, you know, the organic form of nitrogen converting into inorganic form. So the nitrogen cycle will be happening by biological nitrogen fixation, ammonification, nitrification, denitrification process are there. So in the nitrogen cycle, biological nitrogen fixation and the nitrification steps are very, very important. So these biological nitrogen fixation by important soil microorganisms like Rhizobium, Azotobacter, Azospirillum, Beijeringia, Frankia type of nitrogen fixing soil microorganisms are involving. So all these microorganisms are involving in the nitrogen fixation process by symbiotically or asymbiotically. They will convert the nitrogen gas into ammonical form with this reaction. So first nitrogen fixing microorganism biofertilizer, that is Rhizobium Biofertilizers. So Rhizobium is a bacterium, soil bacterium. It is a gram negative bacterium, rod shaped bacterium. So it is living in the soils, rhizosphere soils, but it will not fix the, it does not fix the nitrogen in the free living conditions. It will fix the nitrogen when it is symbiotically associated with leguminous plant roots. So when this association is successful, that time the nitrogen fixing bacteria Rhizobium, it will able to fix, form the root nodules and fix the nitrogen into ammonia. So these are the this is the bacterium Rhizobium. This is the Rhizobium bacteria on yeast extract mannitol agar medium. These are the root nodules of the leguminous plant roots. These are the pink color indicating the leghemoglobin pigment present in the root nodules. So the Rhizobium is living in the root nodules, fixing the nitrogen into ammonia. So this ammonia utilized to the root plant growth and metabolism. Almost this Rhizobium contributing 50 to 100 kg nitrogen per hectare per year. So this Rhizobium and legume symbiosis is there. So this is the important soil process in the Rhizobium and leguminous symbiotic relationship. One mutualistic understanding between leguminous plant roots and the Rhizobium. So this Rhizobium roots are releasing the root exudates like flavonoid compounds. These flavonoid compounds are attracting the Rhizobium, expressing the root nodulins, genes that towards the rhizosphere of the root nodule leguminous plants, so that the Rhizobium movement is there. So this Rhizobium will infect the root hairs of the leguminous plant roots and form the root nodules and fix the nitrogen. So this is the this is the process of root nodulation process in the leguminous plant roots. So cross inoculation groups of the Rhizobium. So this Rhizobium biofertilizer suitable for all leguminous plant roots. So one reminder is there. We have to maintain the host specificity of the Rhizobium biofertilizers, because of it is showing host specificity, particularly like regular Rhizobium leguminous crop. It is associated with a particular strain of the Rhizobium. So when we are isolation of any Rhizobium from particular crop, so it will that strain may be utilized to particular crop only. So that time only that host specificity will be successful and nodulation will be more and nitrogen fixing capacity will be enhanced. Here 7 types of cross inoculation groups are there. So here fast growing types are there, slow growing types are there. So fast growing types are means the Rhizobium strain will be under in vitro conditions, laboratory conditions. The growth generation time is less than 6 hours. slow growing types, the generation type of that Rhizobium strain will be more than 6 hours. Here, Rhizobium meliloti suitable for alfalfa, Rhizobium trifoli, clover group, Rhizobium leguminosarum pea group, Rhizobium fascioli, fasciolus, that is beans crop. So slow growing types, Rhizobium lupini, lupinus group, and Rhizobium japonicum or Bradyrhizobium japonicum suitable for soybean crop. Rhizobium species, cowpea group, that is suitable for cowpea, groundnut, chickpea, vigna, fasciolus, and sunhemp. So these are the cross inoculation groups are important thing for Rhizobium biofertilizer application. So nitrogen fixing capacity of the different leguminous plants here mentioned, alfalfa, pea, beans. Here it is there, alfalfa crop 100 to 300 kg nitrogen per hectare. Like this pea and beans, clover, chickpea, peanut. Here the nitrogen fixing capacity is there. So almost legume based cropping system may be successful. That is suggesting for a farming community. It is very, very useful to the improvement of organic matter in organic farming practices. Legume based cropping system is very better choice. Growing off season, seasonal break, we will cultivate a diancha or sesbania in a green manure crop. So it will contribute the organic matter, contribute the carbon, improve the carbon content, and also supplementing the nitrogen. So farmer may reduce the application of nitrogen, further nitrogen to the succeeding crop. So next, Ajospirillum biofertilizers. This is also one of the nitrogen fixing biofertilizer. It is these biofertilizers are associative nitrogen fixing microorganisms. Associative. Generally, primarily, these organisms are living in the soil, rhizosphere, and intercellular spaces of the root cortex of the plants, generally belonging to Gramineaceae family. So this is nitrogen fixer, and also it contribute, it promote, it growth promoting substance releasing bacteria like indolestic acid and gibberellic acid. These bacteria are producing and promoting the plant growth. Some strains of Ajospirillum also, they are disease resistance and drought tolerance also. So Ajospirillum suitable biofertilizer for especially paddy, sugarcane, cotton, and chili crop. It is successful. And Ajospirillum fixes 10 to 30 kg nitrogen per hectare in paddy, and 10 to 20 kg nitrogen per kg per hectare in other crops per season. So it is not host specific, but preference will be with the C4 plants. So in Indian conditions, Ajospirillum lipoferum, Ajospirillum braziliensis strains are famous for biofertilizer preparations. Next, nitrogen fixing biofertilizer, that is Azotobacter. So it is free living nitrogen fixing biofertilizer. It is living in the free condition in the rhizosphere area. It widely used for vegetables and flower gardens. It prefers more organic carbon content for effective nitrogen fixation. So that's why it is mainly suggested to enrichment with FIM or vermicompost for application of Azotobacter biofertilizers. It also producing plant growth promoting hormones like indolestic acid and gibberellic acid and cytokinins, and producing vitamins also to promote the plant growth. So it can tolerate temperature stress because of some strains like Azotobacter, Vinylandi, Azotobacter, Crococum strains are producing cyst structures during their life cycle. So these cyst structures are resist for desiccation and drought tolerance, and also producing exopolysaccharides. These exopolysaccharides are useful for salinity tolerance and drought tolerance and heavy metal tolerance also. So these are the other types of nitrogen fixing biofertilizer, particularly paddy crop, low-lying water cultivated paddy crop, blue-green algae or cyanobacteria. Next one is Ajola. I'm audible, sir, clearly?
Yes, sir. Yes,
Yes, sir.
So Ajola, you know, this is the water fern, aquatic fern plant. It is a live nitrogen manufacturing factory because of Ajola leaves are harboring Anabina ajole, one cyanobacteria, Anabina ajole. It is living in the leaf cavities of Ajola. So this Ajola generally using as a biofertilizer, nitrogen fixing biofertilizer in paddy fields. And more than 40 to 60% nitrogen can be supplemented when Ajola cultivated as a dual crop with rice. So it is also used as a livestock feed material, weed control in paddy. So blue-green algae. So Ajola, in Indian conditions, Ajola pinnata widely using as a biofertilizers for paddy crop and also using livestock feed material. And blue-green algae, Nasdaq and Anabina widely cultivating as a blue-green algal biofertilizers for supplementing nitrogen in paddy field. So generally, naturally inhabited in the paddy fields, and also strains are developed. Different strains are there. Nasdaq and Anabina, it will be promoting as a powder-based biofertilizers in paddy fields. So it is photosynthetic bacteria. Cyanobacteria, it is fixing the nitrogen. So this is the filamental structure. In between filaments of this structure of the bacteria, a heterocyst, one heterocyst thick walled cells are there. So these heterocysts are fixing the nitrogen fixation process because of other cells are photosynthetic cells. So nitrogen fixation process is not possible. So specialized structures also for nitrogen fixation. These are the heterocysts. So this is also contribute to 40 to 50 kg nitrogen per hectare in paddy fields. So Nasdaq and Anabina. So in IRA center is there, Center for Conservation and Utilization of Blue-green algae. So they will supply the cultures of these blue-green algal biofertilizers and also supply of biofertilizers of blue-green algae. And next coming types of biofertilizer, phosphate solubilizing biofertilizers. Phosphate is applying more as a single superphosphate, diammonium phosphate by farming community. So whatever these phosphates are applied, this majority of the phosphate is fixed with other micronutrients like zinc, aluminum, iron, and calcium based on the soil reaction, soil pH. So these fixed phosphorus, inorganic phosphorus, which is insoluble to the plant roots. So this insoluble form of phosphate is solubilized by the bacteria or fungi by the releasing of acids like inorganic acids or organic acids. So that time the chelate phosphate ions are chelating and other phosphates are releasing. So that other phosphates are easily solubilized by the plant roots. So different types of mechanisms are there for phosphate solubilization by bacteria, phosphate solubilizing bacteria. So main mechanism is organic acid production or chelation of cation bound to the phosphates, and inorganic acid phosphate or exopolysaccharide production, and carbonic acid production and H2S production. So these are the different mechanisms by different bacterial species genera for phosphate solubilization. Almost 25 to 30 kg P2O5 may be solubilizing and availability by phosphate solubilizing bacteria per hectare per season. And phosphorus mobilizing biofertilizers. So example for phosphate solubilizing biofertilizers are Bacillus megaterium, Pseudomonas striata, Pseudomonas putida, and Bacillus polymixa, Bacillus amyloliquefaciens, Penibacillus, and phosphate solubilizing fungi, Aspergillus avamori. And phosphorus mobilizing biofertilizers. Example for phosphorus mobilizing biofertilizers that is important to the mycorrhiza, that is Mycorrhizae biofertilizers. Mycorrhizae is a fungi, biotic fungi. It is required live plant roots for infection. It is one symbiotic relationship, mutualistic understanding. The mycorrhiza, it will infect the fungi, it infect the plant roots, and it will cover the plant roots and also penetrate the mycelial structures into root cortex area, that are vesicles and arbuscules. So these arbuscules are transporting the phosphorus from rhizosphere area to longer places from the root zone to root cortex area. So like this mycorrhiza fungi interaction is there, and fungi will get benefited from the plant roots plants by getting carbon source and habitat. So these important thing are mycorrhiza. Broadly, two types are there: ectomycorrhiza and endomycorrhiza. So ectomycorrhiza is generally inhabitant of woody trees of forest, and endomycorrhiza, that is WAM. Generally, all crops are infected with the mycorrhiza. So indigenous mycorrhiza is there. So we can apply this exogenously with the best strains of mycorrhiza like glomus and Scutellaspora, Acalospora as a biofertilizer formulation. So we will improve the rhizosphere area with mycorrhiza, and phosphorus supplementation and transport will be enhanced. So this mycorrhiza fungi penetrates the cortical cells of the roots, increase the surface area of the roots, displace the absorption equilibrium of the phosphate ions, which increases the transfer of phosphate ions. They transport phosphorus and other nutrients from the deeper layers of the soil and deposit in the plant roots. So stimulate the metabolic process. Arbuscules absorb the nutrients into the root system, and they protect the crop from the water stress. They protect the crop from the root pathogens. So this is the mycorrhiza infection process. Sir, please mute your audio. So this is the mycorrhiza. This is the chlamydospore in the soil. So this plant root structure, the mycorrhiza spores, it is germinating and forming mycelial network. So hypal network, it is forming. It is penetrating into the root cortex area and forming arbuscules and vesicles. So these arbuscules are connected to the from the deeper layers of the root portion. So available phosphorus, it is there, and micronutrients are transporting towards to the root cortex region. So this is the difference between a vesicular arbuscular mycorrhiza and a ectomycorrhiza. So ectomycorrhiza, forest trees, it is the mycorrhiza, it will inhabitant on the root surface area and only hartignate is forming. So whereas vesicular arbuscular mycorrhiza, it is forming surface mycelium and also penetrating the mycelial structures into the root cortex region. This is the difference between ectomycorrhiza and endomycorrhiza. So next type of biofertilizer, potassium releasing bacteria. So generally potassium releasing bacteria, potassium in the soils generally occurs mostly as a silicate minerals which are inaccessible to the plants. So these minerals are made available by the bacteria, that is potassium releasing bacteria, because of acid releasing capacity, same like phosphate solubilizing bacteria. So these microorganisms solubilize the silicates by producing organic acids. These organic acids causing the decomposition of silicates and help in the removal of the metal ions, thereby make the available potassium in the soil solution. So that time plants will absorb the potassium easily. Next, plant growth promoting rhizobacteria, that is so plant growth promoting rhizobacteria, generally these are the formalized growth, these used as a plant growth stimulants and biofertilizers and biocontrol because of these bacteria multifaceted mechanisms are there. So during 1981, Cloeper and Scroth they coined the terminology PGPR. They conducted experiment with Pseudomonas fluorescens in potato crop. So that time they found that blight disease is controlled and also plant growth is promoted with this bacterial formulation. So that time they had given the definition. So these are the bacterial root colonizing bacteria which are promoting the plant growth potentially by direct and indirect activities. So these are the mechanisms of the PGPR, direct mechanisms like nitrogen fixation, phosphate solubilization, and phytohormone production and siderophore production. And indirect plant growth promoting activities like lytic enzymes, like glucanases, cellulases, pectinases enzymes by these bacteria, and antibiotic production. These antibiotic production kill or suppress the pathogenic fungi and bacteria in the soils, disease causing bacteria. This induced systemic resistance like when we are treating PGPR, the plant gets immunized with individual systemic resistance. Naturally, also plant immunized with systemic acquired resistance with pathogenic plant interaction and PR proteins expression. And exopolysaccharide production also is there. Exopolysaccharide production by PGPR bacteria, it will useful for drought tolerance and heavy metal tolerance and salinity tolerance. And also some other activity is there, PGPR ACC deaminase activity, that also important PGPR activity under drought conditions. It will mitigate the stress conditions. And other types of biofertilizer inoculants are there. There are compost inoculants, compost biofertilizers. Suppose we are preparing different types of composting methods to decompose the agricultural produce, and also recently a lot of so many farmers are using burning the crop residues. So these crop burning may be harmful to the environment, causing the pollution and also decreasing the microbial content in the soils. So these all waste maybe residues may be useful to make the compost by utilizing different compost degrading microorganisms like Bacillus, Aerobasidium, Aspergillus, Chaetomium, Penicillium, Pseudomonas, and Trichoderma species. So waste decomposer is one of the consortia of microbial organisms. So this will composting useful as the compost making and also enriching the nutrients. Like Pusa decomposer also is there. Like this, this we will prepare the consortium group of microorganisms to decompose the residue and making into compost. So it will reduce the time of composting, general composting process, and also enriching the more nutrients in the compost. Why? What are the reasons for low adapting the biofertilizer utilization by farming community? Because of inconsistent response is there, and low response compared to the chemicals, and low organic carbon in the Indian soils, and high ambient temperature, and short shelf life of the different formulations, and being low cost, low trade margin is not attractive. Poor quality and absence of temperature controlled delivery systems, and low support and incentives compared to chemical fertilizers. So some interventions are made to overcome the problems because of so like the introduction of sterile liquid biofertilizer inoculants or novel inoculants, capsules, or other types of inoculants. Effective quality control mechanism is required. Better formulations with a longer shelf life and temperature tolerant formulations are required. Introduction of consortia cultures, increased financial support for setting up of sterile inoculant production facilities. So these are the some interventions made to overcome the problems or concerns. So next important thing is mass production of biofertilizers and application of biofertilizers in organic farming. So before mass production, some different biofertilizer production centers or organizations or institute, they have to maintain their own isolates, characterized isolates for biofertilizer production. Some organizations or some biofertilizer production centers, they are procuring the strains from culture preservation centers and characterizing, and they are conducting the trials whether that strain is efficient or not, and after that they are preparing the formulations. So these are the different isolation procedures for different biofertilizer isolates. So different media for standard media for biofertilizer isolates, isolation and mass pulverization. So generally, so for Rhizobium bacteria, mass production, isolation and mass production, generally yeast extract mannitol agar generally media broth will be using yeast extract mannitol with congruent media agar for isolation generally using. And Azotobacter bacteria, so we are generally suggesting Jensen's agar media or Buck's agar media or Aspie's agar media or vaccine number 77 media for isolation and mass multiplication. Also, these are the media suggested without agar. So azospirillum, so nitrogen free bromothymol blue medium, semi-solid medium for isolation, and broth, same kind of broth suggested for mass multiplication. Gluconoisobacter diatrophicus bacteria. This is nitrogen fixing bacteria for sugarcane crop. So media for this crop is LGIP medium. And blue green algae. So generally it required inorganic nutrients only because of it is photosynthetic bacteria. So fox or Bristol medium generally suggested. And phosphate solubilizing bacterial biofertilizer growing media, that is Picovascia agar. It is phosphate enriched media, that is tricalcium phosphate enriched media. Whereas potassium releasing bacteria, that is Alexandrov media, that is inorganic potassium aluminum silicate enriched media. Zinc solubilizing media required mineral salts medium with enrichment of any insoluble inorganic zinc medium like zinc oxide or zinc carbonate or zinc phosphate. While silicate solubilizing bacteria, modified Bunt and Rovira medium is required. Sulfur oxidizing bacteria, Starkey broth and NCl broth and thiosulfate medium. Compost inoculants, aspergine medium or Hans medium generally using. For methylotrophic bacteria, pink pigmented methylotrophic bacteria, mineral salts medium, it is used suggestible. Generally, commonly Rhizobium, Azotobacter, Azospirillum, and phosphorus solubilizing bacteria, potassium releasing bacteria, and zinc solubilizing bacteria producing by so many biofertilizer centers. So this is the technique generally for isolation of any bacteria from soils. We need to prepare serial saline blanks or distilled water saline blanks for isolation of the bacteria from soil samples. So we are taking the 1 gram or 10 gram, 1 gram soil sample. We are mixing with 9 ml of saline water, sterile saline water, properly mixing, properly vortexing it, and again transferring in 1 ml from first dilution blank to second dilution blank. Like this, again rotate, vortexing. So like this, up to 10 power minus 7 to or 8 dilution blanks we are preparing, and we are reducing the concentration of the soil sample. So after serial dilution technique, we are taking the 0.1 ml into the suitable media Petri plates, and after spreading, please, hello sir, please mute your audio. So this after plating the 0.1 ml on suitable media Petri plates for specific bacteria, for specific media plates we can use. So here spreading, after incubation we will get the colonies. So we will pick up the one isolated colony from this plate, and we have to characterize, and we will see the under microscopic observation, and we will characterize, and molecular identification we will get the details of the strain. So these are the plating methods, the spread plate method and pour plate method. Generally this is the basic information, you might be know about these techniques. For Rhizobium bacteria isolation from the root nodules, so this is the procedure. So media is yeast extract mannitol agar media. So first to collect the root nodules from the your crop samples. So stage is root nodule flowering stage. We will select pink color nodules. We will pick from root nodules. So collect the total root system from the field. Clean the root system and select the pink color nodules, and pick the separate the root nodules from the plant, and surface sterilize with mercury chloride and alcohol, and crush the root nodules by using sterile pestle and mortar, and take this suspension, this crushed suspension into serial dilutions. Make the serial dilution, and take the suspension from the dilution on specific media, that is congruent yeast extract mannitol agar media. So incubate the petri plates for 30 to plus or minus 2 degrees centigrade for 2 to 3 days. So after incubation, we will get the rhizobium colonies on the plate. So this is the procedure. So we will collect the pink color root nodules, surface sterilize the pink color root nodules, and pagel and mortar these root nodules. After that, take the 0.1 ml sample into the this petri plate, sterile media petri plate. After incubation, we will get the rhizobium colonies. So rhizobium colonies are white color, translucent colonies on the red color media plate. So while other red color colonies are contamination. So this is the colony growth of rhizobium on yeast extract mannitol agar medium. So here you can see white color colonies are white color mucoid colonies, translucent colonies are rhizobium colonies, whereas red color colonies, that is agrobacterium, there are other than rhizobium, these are the contaminated colonies on the yeast extract mannitol agar medium. So the petri plates media, it is a pink color, but rhizobium will not, does not absorb the pink color. Other contaminated colonies are absorbing the pink color. So this is the visible characterization of the rhizobium colony. So testing of the nodulation efficiency of the rhizobium, whatever your isolated rhizobium, whether it is in vitro condition. Sir, please mute your audio, sir. So these in vitro conditions and in vivo conditions, we have to see the efficacy of the rhizobium strain, whether it is fixing the root nodules with the cross inoculation groups. After efficiency testing, we will go for mass production of the rhizobium bacterial biofertilizer. Next, Azotobacter biofertilizer. So sample is rhizosphere soil sample. Same like we will take the 1 gram sample into first dilution blank. We will prepare the dilution serial dilutions and fix. What is that? We will take the dilution sample and plating on Jensen's agar media, and the next step is incubation. So after incubation, we will get the colonies. Initially 2 to 3 days, we will get a transparent gum type of colonies. After incubation, 5 to 7 days. So the Azotobacter chlorococcum, it is giving bluish, brownish black pigmentation. Azotobacter vinylandi also same like that. Azotobacter paspali, yellowish pigmentation. Azotobacter beijingia, light greenish yellowish pigmentation. So with the cultural characteristics, we will identify the bacteria, and a microscopic also staining, also gram staining procedure also, we will see the structure of the bacteria. Next, Azospirillum biofertilizer. So Azospirillum biofertilizer isolation media is nitrogen-free semi-solid medium, for isolation. Sample is here. So Azospirillum is a associative symbiotic nitrogen fixing biofertilizer. It is living in the root surface area. That's why we are taking root hair samples for isolation. We are dipping the samples in the semi-solid medium by following most probable number method technique. So if after incubation 2 to 3 days, it is showing on this semi-solid medium, semi-solid subsurface white pellicles are forming by this bacteria. So these subsurface pellicles are indicating the bacterial growth, Azospirillum growth. Because of Azospirillum is a microaerophilic bacteria, it required low oxygen conditions for growth. So that's why it is growing on a subsurface on the media, so in the test tube, semi-solid medium. So it is here. This is it is control tube. So here control, it is negative for Azospirillum. So these are the positive. So this is the Azospirillum growth on nitrogen-free medium. It is bromothiamol blue medium. Phosphorus solubilizing bacteria for isolation, we are taking soil sample and using the media, phosphate enriched media. So for we need to enrich with insoluble tricalcium phosphate in the medium. So for isolating the phosphate solubilizing bacteria. So same like the serial dilution technique, we are taking 1 gram soil sample, we are diluting, and after that dilution we are taking 0.1 ml into the petri plates, that is Mycorrhizae agar medium. After incubation of 3 to 5 days, we will get the colonies like this phosphate solubilizing bacterial colonies. So the colonies are clearing, solubilized zones are there on this Mycorrhizae agar medium. That zones are indicating the bacteria is phosphate solubilizing bacteria. So we will measure the zone, and also we will see the test efficacy of the bacteria, how much phosphorus is availability in the liquid Mycorrhizae broth medium also. Next, zinc solubilizing bacteria isolation. Same like phosphate solubilizing bacteria isolation, we are using bunt and roviera medium. Bunt and roviera medium or mineral salt medium for isolation of the zinc solubilizing bacteria with any compound like zinc oxide or zinc phosphate or zinc carbonate or zinc phosphate you can use. So this is the zinc solubilizing bacteria isolation by using mineral salts medium. It's providing zinc as a source. So next, mycorrhiza also isolation and production is different from the other bacterial biofertilizers because if it is a biotic fungi, it required live plant roots for mass multiplication. For isolation of mycorrhiza from soil samples, this is the procedure. Mycorrhiza infection, whether the mycorrhiza is infected with your plant roots or not, we will see this with this procedure. So first we will take the root sample, collect the plant root system, and take the roots, fine roots, and cut the root pieces into small root bits, 1 centimeter bits, and dip the root bits with formalin acetic acid solution. After that, wash the root system, and again treat with 10% KOH for 30 minutes at 40 degrees centigrade. After the next step is pour the rinse the root system with water and 2-3 times repeatedly. Next, these roots are crushing and making into crushing with a sterile root needle, and also add Trypan blue 0.0% Trypan blue for 24 hours at room temperature. So this is the staining procedure, and also add lactoglycerol to remove the excessive stain Trypan blue from the slide. So next thing is you just visualize under this slide to be visualized under microscopic 40x. So mycorrhiza infection, it is like this. So here arbuscles are there in the root cortex region. So that your plant will be infected with mycorrhiza. So these roots also used for further mass multiplication procedure. Otherwise, you can isolate the spores from the soil sample. You can propagate the mycorrhiza. Again, these spores to be infected with the plant roots, and you can follow the mass production technique. Hello, madam. Hello.
Hello,
Pandya
sir.
madam. Pandya madam. Hello. Sir, sorry for the inconvenience, sir. I will take 5 minutes break, sir. I will join again, sir.
Yeah, sure, sir. No problem, sir.
Yes.
Okay. Yeah.
So next, mycorrhiza spores isolation from the soil sample. So this is also important method is there. Wet sieving and decanting method is there. So this from this wet sieving and decanting method, so mycorrhiza spores will be harvested. So take 100 grams soil sample from the 100 grams rhizosphere soil sample and put in dip in mix with 500 ml of water. So after mixing properly mix it and wait for 30 minutes. So proper settlement of the heavy particles and take the supernatant soil solution into sieves. Different types of sieves are there. So these sieves are filter the mycorrhiza spores. So this whatever sievate is there, filter the sievate, collect the sievate into the petri plate with a dispenser and these collected spores particles to be visualized under microscopic observation. So we will see the microscopic viable spores of the mycorrhiza and we will take that spores for another inoculum purpose under maintenance. So this is the procedure. So these are the mycorrhiza spores. So mycorrhiza characterization also possible with the characteristics of the different mycorrhiza spores are mentioned here. So glomus, what are the spore characteristics? Glomus, acalospora characteristics, scutellaspora characteristics, and gigaspora, acalospora, different VAM spores are mentioned here, characteristics. And coming to mass production of the biofertilizers, sir, audible, sir? Clearly audible, sir?
Yes, sir. Audible is there, no problem.
Yes, sir. Yes.
So before going, so state agriculture government, state agriculture department, and universities and other private organizations, private industries are commercially producing biofertilizers. So any biofertilizer production unit, so generally follow the guidelines of fertilizer control order guidelines. So unit of manufacturing and selling license is required. So manufacturing license and selling license are required. So as per the government FCA requirement, FCA guidelines, state government is commiserated, they will issue the license. This is valid for 3 years. So once in 3 years, you have to renew the license copy. So any production technique of this, any commercial production techniques, some steps are required before commercial production, before producing bulk amount, more amount. We have to select a better strain. So like ability, so criteria for strain selection and N-fixing and PK solubilizing bacteria. So take better strain, competitive strain, first thing, whether they are in vitro conditions and in vivo conditions, they are fitting, their test efficacy are showing, that strains to be selected, and these strains to be multiplied, and you check whether the strains are able to surviving in different soils also. And the next step is media preparation, sterilization and fermentation process, and the culture to be multiplied in levels. Primary level using saccharin flask, secondary stage of multiplication in the fermenters.
Excuse me.
Hello.
Sir.
Tell me, sir.
Is it compulsory to check the strain available in the soil, because the soils are different?
Yes, sir. Generally, suppose different agroclimatic is their regions are there, sir. Suppose you are getting strain from other climatic regions, maybe it will be able to viable in our area to be checked, sir.
Is it practically possible, sir, for a person who
is not possible, sir, but better development of commercial production. So before we are making into commercial fermentation, that strain to be checked, efficacy is there or not, whether it is persisting in the soils. That checking means better option, sir, for working of a strain effectively.
That's a good parameter, but unfortunately, we don't have any truthful
yes,
label
yes.
by any manufacturers.
Yes, sir. Yes, sir. Sorry, sir. I will discuss this discussion part will be there.
Collection da. Hand over பண்ணனும். இவங்கிட்ட எல்லாம் வாங்குனாங்க இல்லையா? அதை கொண்டு வந்து எங்க கிட்ட ஹேண்ட் ஓவர் பண்ணனும். அதை எல்லாமே ப்ராப்பரா இருக்கா செக் பண்ணி வாங்கி வைக்கணும். வாங்கி வச்சு
ஹலோ ஹலோ சார்.
எல்லாரும் டிஸ்போஸ் ஆனாங்க. மேடமும்
ஹலோ சார். Sir, I will discuss the things in discussion part, sir.
எல்லாரும் இருந்து அந்த சீல் பண்ணி கொடுத்துட்டு வெளியே வந்தோம்.
Sir, please. Hello, sir. Please mute your audio, sir. So, in case of Rhizobium, Azotobacter, PSB, KSB, molasses could serve as a superior source for growth and multiplication. Different source of media we can suggesting. So already I mentioned different types of media for different inoculants. So media preparation, sterilization, fermentation, generally microorganisms to be multiplied for biofertilizer production may be grown in batch, continuous culture and in fed batch fermentation, generally following batch fermentation in all commercial production centers. So in batch fermentation, it is closed type of fermentation. Initially, we are using production media, sterilizing and adding 1% to 5% inoculant and incubating with sterile condition, optimum conditions in the fermenter, and checking the quality control and using stationary maximal rate culture to the preparation of the inoculant, whether it is a carrier based biofertilizer or liquid based biofertilizers. So this is the workflow for mass production. So prepare appropriate media for specific to bacterial inoculant in required quantity. Inoculate with a specific bacterial strain aseptically. Incubate at 30 plus or 2 degrees centigrade for 5 to 7 days in rotary shaker. Observe growth of culture and allow the population to reach around 10 power 10 or 10 power 11 as starter culture. Under optimum conditions, this population level could be attained with 2 to 3 days per PSB, KRB, for Rhizobium 4 to 5 days, for general KSB, KRB, PSB 4 to 5 days, Rhizobium 5 to 7 days, or 6 to 7 days for Azotobacter. So all this above media is prepared in large quantities in fermenter. So after sterilization, the sterilized and cooled well and media in fermenter is inoculated with a log phase of culture grown in large flask. Usually 3 to 5% of inoculum is sufficient. Cultures are grown in fermenter providing aeration, continuous stirring, and other favorable conditions. Broth is checked for population in inoculated organisms. Broth is harvested with a population load of 10 to 10 power 11 cells per ml. So fermentation, final culture is prepared in this step. Fermenter is operated with 2 by 3rd portion of its capacity with proper aeration and agitation. That a headspace volume is required. Sterile medium in the fermenter is inoculated with seed culture, and aeration will be supplied with the filtered filters, air filters. Fermentation is done for 3 to 5 days, 3 to 7 days, until population reaches 10 to 10 powers 10 or 11 per ml. The broth is allowed to age. During this process, microorganisms secrete some acids in the solution, due to which pH decreases very much. So pH is maintained by adding NaOH or H2SO4. So this is the production of carrier-based biofertilizers. So we are collecting them, we are isolating the bacteria, characterizing, and directly procuring the mother culture, these strains from the culture preservation centers. We are checking the purity. Again, flask level culture we are inoculating, and this flask level, big flask level culture, and next inoculating into fermenters, big fermenters, and providing optimum conditions and checking the quality. And simultaneously, we are preparing the carrier material and sterilizing the neutralizing the carrier material with the calcium carbonate. And after neutralization, we are mixing the carrier material with broth of the bacteria, and 1 day curing. After mixing we are curing for 1 day. Next, that is after mixing, after curing 1 day, next packing with labeled polythene, standardized polythene bags. So these polythene bags, this is 1 kg, half kg. See, these bags are supplemented to the distributed to the farmers. So carrier-based biofertilizers are some drawbacks, disadvantages is that because shorter shelf life is there, 3 to 6 months, and sensitivity to temperatures, and more chances of contamination, and unavailability of good carrier in local area, bulk sterilization problems in terms of economy and facilities, poor moisture retention capacity, labor intensive, and problems for proper packing. High transport cost is there. It takes more time for quality control. Dosage contradicted to farmers. Next, liquid biofertilizers production. This is the same method of carrier-based biofertilizer. Modification is there. Here, no need to add any carrier material of preparation and processing. Here, directly broth itself we are using quality broth, but we are adding some liquid cell protective materials like cell protectants or additives, or like glycerol, polyvinyl pyrrolidine, or iron EDTA, or trehalose, or sucrose, or glucose compounds to increase the shelf life of the product. Before harvesting, we are inoculating these sterilized compounds. and next we are packing into bottles and distributing to the farmers. So liquid stabilizers are cell protectants like trehalose, glucose, iron EDTA, PV polyvinyl pyrrolidine, glycerol, mannitol. Generally, we are using. So advantages is there with the liquid biofertilizers because of these products, liquid biofertilizers giving shelf life 12 to 24 months, and the contamination problem is very less, and no loss of properties due to storage up to 45 degrees centigrade, and greater potential to fight with the native population, easy identification by typical fermented smell, better survival on seeds and soil, very much easy to use by the farmer. High commercial revenues are there with the liquid formulations. So coming to NIPHM technology, on-farm production technology of biofertilizers. So generally constraints are there in biofertilizer adapting by farmers because of availability is no idea about availability, and how to apply these biofertilizers, how to recommend for different crops. So farmers are lacking this type of knowledge, and also quality biofertilizers formulations are missing in nowadays. So that's why NIPHM is developed this on-farm production technology, on-farm production of biofertilizer technology to the farmers, especially small and marginal farmers. They can use this, adapt this technology. They can produce their own biofertilizers by this standard procedure. So aims of the biofertilizer, this on-farm production technology, because of to ensure the quality of biofertilizers in inoculum in large quantities at the farmer's field. Very little cost technology is required and easy to establish. So these are the materials required for on-farm production technology of biofertilizer: LPG cylinder, and 14 that is approximately cost around 2,000 rupees to 3,000 rupees, and pressure cooker, 20-liter pressure cooker, and gas stove, 2-burner gas stove, and inoculation chamber, wooden inoculation chamber, and sterilizable vials, 5 ml vials, and miscellaneous items like waste glass bottles, spirit lamp, jaggery, cotton, etc., and mother culture. So approximately 25,000 to 35,000 rupees are cost for establishing this biofertilizer production on-farm production laboratory for production of biofertilizer. So 2 minutes, sir. Swapna, sir, my room bag lo water bottle untadi kodhi OP sir ni sir తీసుకున్నారు urgent గా రా Swapna, water bottle ఉంటుంది second zip లో ఉంటుంది. So these are the materials are required for on-farm production: gas cylinder, pressure cooker, gas stove with 2-burner wooden inoculation chamber, sterilizable vials, miscellaneous items like bottles, waste glass bottles, spirit lamp, jaggery, cotton, and mother cultures. So mother cultures, farmers can get mother culture from any culture. This biofertilizer production center, or generally NIPHM is providing to the issuing to the mother cultures to the farmer who are interested to produce these adapting biofertilizer production technique, and also who are getting training at NIPHM, 3 days farmers training, we are issuing the mother cultures. So this is the procedure on-farm production technique of biofertilizers. So this is here Rhizobium bacteria biofertilizer production mentioned. So this is a technique similar, say common for all bacterial biofertilizer isolates. So simple technique. So this is suggested for farmers at their farmers fields, their own production. Here, first thing is jaggery media. We are suggesting to multiply the bacterial biofertilizers. So first thing is farmers to prepare jaggery media by adding 10 grams jaggery, 1 gram common salt in 1 liter mineral water. Properly mix it well. After mixing properly, transfer this jaggery media into cleaned waste glass bottles. So here waste glass bottles are there. So fill the bacteria 1/3 portion and 250 ml like this in 1 liter plus 1 liter glass bottles or liter glass bottles. So put the cotton plugging and wrap with paper and tie the rubber band. So like this 4 to 5 bottles you can prepare. And the next step is sterilization. Here cooker. So cooker is suggested for this production. Cooker farmers level. This is the sterilization equipment. So and also gas burner, gas stove, gas cylinder and gas stove we are using for sterilization of the cooker. So here gas stove, gas cooker. It is 20 liter cooker. Hello. Anna security guard ki cheyandi anna, main gate security guard. Hello. Okay. So this is the cooker for sterilization of your jaggery media bottles. So take some water in the cooker and put your all jaggery media bottles inside of the chamber and close the lid and sterilize up to wait for 40 minutes. After 40 minutes, the cooker will be sterilizing your jaggery media bottles and wait for cooling, 0 pressure, and take out your bottles and wait for room temperature. Next step, this inoculating of mother culture into jaggery media, sterilized jaggery media bottles. So this inoculation purpose, we are suggesting use wooden inoculation chamber. So this wooden inoculation chamber is like this. So wooden inoculation chamber 40 centimeter size is there. So this wooden inoculation chamber facilitated with a glass wooden transparent door. It is flexible for opening and closing type, and it is facilitated with the 2 holes for inoculating the chamber, inoculating the mother culture. So this chamber, these holes for inserting your hands and inoculating the mother culture into jaggery media. Before inoculating to the mother culture into the jaggery media, clean the surface, clean the this workbench of the chamber with a disinfectant solution. Put the spirit lamp and jaggery media, sterile jaggery media bottles and mother cultures. And some farmers they are facilitated UV light and normal light also in this chamber. So UV light may be used before inoculation. Treating for 10 minutes, the chamber will be sterilizing, and normal light may be useful for during inoculation step. So these sterilized bottles are keep at inside of the chamber and wear gloves and mask and surface sterilize your hands and inoculate the mother cultures 1 ml culture to be inoculated into these jaggery media bottles. near the spirit lamp. So each mother, suppose you want to produce one type of biofertilizer, take one mother culture, inoculate this mother culture into all bottles. Suppose you want to produce one type of biofertilizer, 1 ml of specific type of mother culture to be inoculated into that bottle, specific Jaggery media bottle. After inoculating, label with the type of that biofertilizer name and date. After inoculating, same cotton plugging, wrapping with paper, and next step is incubation. So incubate the bottle, not required 3 to 4 days, 5 to 7 days is not required. So generally bacteria, just 1 or 2 days is sufficient for multiplication of the biofertilizer. So during incubation, you just gently shake the flask. After incubation, so biofertilizer will be ready. So this will be useful for application. Immediate use is generally suggested. Suppose farmers want to store this biofertilizer, they can store under cooling conditions, refrigerated conditions. Because of here, farmers does not add any sterile cell protectant or cell like glycerol or PVP. So that's why farmers can immediately after production they can use for different methods like soil application or seedling dip method or any method of application. So this is the on-farm production technique of bacterial biofertilizers. This is our NIPHM fermenter facility. We are producing different types of liquid formulations of different biofertilizers. So next method is the on-farm production of Ajola. So Ajola also we can produce easily. So we will dig the small pit and we will line the pit with polythene sheet and add soil and cow dung and single superphosphate slurry and add mother and maintain the 10 centimeter water level and add mother culture, Ajola culture, small amount of other culture for pit, and we will get within 10 to 12 days we will observe the Ajola culture on the pit. So this can be applied in paddy field after transplanting after 7 days. So within this culture, this hello sir, I will call back sir after 1 hour. Yes sir, yes sir, yes sir. I will call back sir. Actually I'm in seminar. Yes sir, yes sir, yes sir. So every 3 to 4 weeks you have to change your soil and water in the pit. This is the procedure. So this is the Ajola can be used as a nitrogen fertilizer, livestock material, livestock feed material, and mosquito flame and weed control. So next, different bacterial biofertilizer application methods. So before going to application methods, so seed treatment. For seed treatment, generally 200 ml per acre liquid biofertilizer is suggested. So for seed treatment, generally take 200 ml of liquid biofertilizer and take 1 acre required seed material, mix with this biofertilizer and wait for 30 minutes under shade dry conditions. After that, you can sow the seed for seed treatment. So for seedling dip method, generally transplanting crops we can use. So seedling dip method, so 4 to 5 ml of biofertilizer solution, 4 to 5 ml of biofertilizer solution in 1 liter water. Prepare the slurry and dip the seedlings in your this slurry for 20 minutes. After that, you can apply. You can take these seedlings, transplant in your main field area. So these seeds, these roots are dipped in the solution. So the roots are seeds can absorb the solution. Maybe that bacteria will enrich the soil. Soil application, generally 200 ml dose per acre 200 ml of biofertilizer will mix with 100 kg of FIM or vermicompost. So these enriched compost enriched for 10 days. After that, we can apply this enriched compost in your main field application. Final flowering and moisture is required, and at least 5 to 10 days gap is required for chemical fertilizer application. So this soil application, it will improve the soil fertility. For drip irrigation, liquid biofertilizers are suggested. For 100 liters of fertigation tank, you can apply. You can add 200 ml of liquid formulations and properly mix it and irrigate the field with drip channels. So mycorrhiza on-farm production also method is there. So mycorrhiza on-farm production. Mycorrhiza on-farm production. Generally, mycorrhiza on-farm production not possible by jaggery media, any other media. It required live plant roots for mass multiplication. So that's why NIPHM developed one part method for mycorrhiza on-farm production. So this part method is very easy, very useful. So, excuse me, madam, can I share any video, madam? Video?
Yeah, no problem, sir. You can
Yes,
share.
sir, you can, sir.